Now, we are very familiar about our pollutions, natural resources. Natural resources like water, air, and also soil. Because of our all anthropogenic activities. Yes. Are you hearing my voice? Hello?
Sir,
Yes,
yes, sir.
sir. Yes, sir.
Can you hear my voice?
Yes, sir. Yes, sir. It's audible.
Yes. Okay. So now, these are all air pollution and also remedy measures. You all know that the air, the pollution in water, air, or soil, which we are deteriorating by doing many anthropogenic activities. If you take air, nowadays the air pollution is increasing all urban cities because of all our anthropogenic or transportation, dumping of waste, burning of waste, and also vehicular movements. All these emissions, we are polluting our quality of air. So we can survive without water and also food, but we cannot survive even 10 minutes without air. We need all the time pure air. So my research area mainly focusing on this air quality and remedies, phytoremediations. That I will come to little later. Then all this air pollution, quality deteriorating, types of air pollution, and also this we will discuss later. A similar way, we are using water. Water is our natural resource. All this industrial effluents, all this sewage effluents, all this due to urbanization and also dumping of all this waste in the urban cities. When rainfall comes, it runs away to the water systems. Wherever these urban areas, wherever the metropolitan cities, in and around you see the water, you can see the more water, but you cannot touch the water. Yes or no? You can see the more water in and around urban areas, but even you cannot touch that water, you cannot use that water. That much of polluting, that much of pollution taking place in and around all our urban cities. You take any cities, all metropolitan cities or major cities or district headquarters, you take this. This is happening all the places. And the sources of water pollution, if you take many areas, many types. And then even soil. Soil we are polluting. This is also natural resource. We are polluting in a lot by using all that synthetic pesticides in the agriculture field. Synthetic pesticide dumping in the agriculture field, and we are deteriorating the quality of soil, and fertility of the soil is decreasing, and crop production is a variation, and the quality of the crops also varying because of dumping all that synthetic material, synthetic organic material, chemical fertilizers, which we are deteriorating the quality of soil also. That means what we are getting freely, soil, air, and water. Totally, we are not believing 100%. You can take the water in the 100%. You cannot drink it the tap water, or you cannot believe the air what we are getting is 100% pure, and the soil also we are not getting 100% quality. Then there is a lot of research going on this air pollution, quality characteristics, and also composition and biological impacts on that. And lot of research going on water pollution, water toxicity, water pollution impacts on aquatic animals, and water pollution on agriculture area. That and all lot of research came and also doing in the large scale. Publications are coming. And soil quality also. Soil quality, soil texture, soil pollution, soil types, everything. Lot of pollution. The publications you have seen, if you see the journals, or if you search the journals or review the journals. My concept is a lot of publications on water quality, soil quality, and air qualities. Then what are the remediations which we are going to start? or what are the remediations we can find biologically or naturally without disturbing by putting other any chemicals. Okay. Though it is a cost effective, it is taking a lot of time and also more economical also that is. So my point is finding the solution in a little way by using natural sources and finding some solution using that natural sources to get that remedies, that is phytoremedies. Phyto means you all know that plants. Okay. Plants are the, is there any plants which are capable of degradation or which are capable of absorbing our gaseous pollutants or which are absorbing the water toxic pollutants or in the soil or in the water. So my area of this research is after observing, after doing all this research on water quality, research on toxicity, research on impact of these pollutants on animal model, then we are stick on on this remediation processes. This today presentation is 3 projects, the research work of 3 projects. One is Department of Science and Technology, DST, one from VGST, Vision Group of Science and Technology, one from ICMR. Combination of these 3 that I am showing now, what are the work which we are doing it now. Okay, you got it? You got my point?
Yes, sir.
Yes, sir. Yes,
See,
sir.
there is a lot of publications on air pollution, water pollution, and soil pollution. I'm not touching that. But what the projects is sanctioned for remediation process by DST, Department of Science and Technology, and our Karnataka government has given VGST, phytoremediation, one project, and ICMR gave 1 crore project, which we are doing by combination of all this. This I am going to present it now today. Okay, I'll skip many places. Okay, you got my point now. Only on remediation process, which sanctioned by Government of Karnataka, sanctioned by Government of India, and also sanctioned by the Department of Science and Technology. That is, now it is running in our department. There's 3 projects which I am running this. So I am the principal investigator to do this projects. Okay, phytoremediation. That is development of low-cost phytoremediation to improve air and soil quality. This is the air. I think this is no need to explain this. Now, at present, all industries you are going to see this much of emissions from industries. This much of smoke emission. That is all. This is what you see. This is all particulate matter. PM10, PM2.5, PM1. If you see that smoke, what you are observing is a combination of these 3 and addition to many gases, air pollutants. This is the percentage of what is that air pollution concentrations. For example, industrial sector 51% we are contributing, remaining agriculture, waste dumping and other sources and also transportation. So this is the percentage wise if you observe the statistics of this air pollution. I am talking about the air pollution now. So air pollution concentration or air pollutants entering into the atmosphere, this is the sectors, industrial sector, agriculture sector and waste dumping sectors, transportation sectors. The maximum contributing from industries which emitting a lot of pollutants into the atmosphere. This is not a new thing. Air pollution is not a new thing. It was started before independence, before BC. So there is a air pollution in earth originated. The man when started using fire for cooking. Okay. And Hippocrates says that 400 BC, father of medicine, for his 80 written medical works, had written about the air pollution prevailing even those ancient times. That means air pollution is not a new, but the level of air pollution is new. The concentration of air pollution which are emitting and releasing into the atmosphere is new. And Edward 1200 AD is the first producing this anti-pollution law, anti-air pollution law in 1273. And other 13,000, another law was enacted in London. So banning the use of coal and prescribing capital punishment for defiance of this law. What are the major sources of this air pollution? The natural sources, naturally the forest fires. Now you can see the California, in and around, pollen and dust, and also some other volcanic eruptions. Volcanic eruption is also major source in the natural way. No one can stop this. And unnatural is motor vehicle exhaust, heat and power generation facilities, industrial process, auto manufacturing, and fertilizing plants, and building demolitions. These are all what the natural sources. Compared to that, unnatural sources is increasing the percentage. That is why Delhi is facing last 3 months the biggest problem of smog, and all metropolitan cities facing a lot of transport emission factors. Okay, so these are this, and you know this episode 1952, the London smog and Los Angeles smog 1954. London smog, Los Angeles smog is a big death of about 4,000 people by accumulation of air pollutants over the London city for 5 consecutive days. London smog and Los Angeles smog is the biggest episodes. It's a combination of sulfur dioxide and nitrogen dioxide, and also a combination of sulfur dioxide and the H2SO4. Okay, so there is, methyl isocyanate disaster, December 2-3, 1984. The methyl isocyanate released into the water, and the more emission of smoke, and lot of people are affected by methyl isocyanate, and nearly still they are suffering some places in this incident. This is the National Ambient Air Quality Standards, which prescribed ISO, pollution control boards. Okay, all these are all the permissible limits should be maintained, but all exceeding in all the places where the urban areas, sulfur dioxide, oxides of nitrogen, particulate matter 2.5, particulate matter 10, and lead and other gaseous pollutants. So these are the concentrating ambient air industrial area, ecologically sensitive area, which prescribed WHO and also ISO and pollution control board. It is the permissible limit. But where is this permissible limit? Exceeding all the places because of all our anthropogenic activities which are running every day, which we are doing, we are contributing this. These are all the standards of arsenic, nickel, and also this method, the last column is what are the methods or methodology to analyze this. That I am in index. What air quality index indicates whether pollutant levels air may cause health concerns, ranges from 0 to 500. Okay, a particulate matter monitoring system that employ tapered element oscillating microbalances, that is TEOM technology to measure the particulate matter concentration. My research mainly focusing on PM, particulate matter. This is the biggest episode, and this is the major contribution. This is the biggest problem in India suffering because of this PM. Okay, this is the calculation, how you are going to calculate the PM level after obtaining from different sources. This is the air quality index, AQI value. You can see this in your Google also, your pollution control boards, they are mentioned in front of the office. 0 to 50 is a good level. Concern, little or no risk at all. If the PM air quality level is 50, up to 50, 50 to 100 acceptable to air quality. And where we are getting this 101 to 150 unhealthy for sensitive groups. General public not likely to accept this. Majority of the places of India, majority of metro cities, always exceeding 101 to 150 in some places. I will show you that chart also. This. So if increasing 201 to 300, okay, is a one very unhealthy conditions where you are observing in Delhi, where sometimes you are observing in even Bangalore also this condition in Chennai and also Mumbai. Many seasons we are getting this result 201 to 300. Some seasons of particular areas. This world now, the WHO releases a new global pollution data on 2nd May 2020. See, where is India? India now first place after India, the China, Pakistan, Egypt, Nepal, South Africa, Mexico, USA. So this is the concentration of the pollutants in India, China, Pakistan, Egypt. The level is given 130, 120, 110. Air quality index levels, global pollution levels in from many countries. Either WHO releases this data on May 2020. This is the 20 worst cities worldwide for air pollution status. See, India, many places getting 317. Okay, and Raipur 268. And even you can see the Gwalior 329. See, air quality 1 to 50. Where is 1 to 50? Where is 150 to 100 or 150? Where is 200 to 300? It is many places, nearly 10 places of India identified more than 200. So this is the chart you will see. Delhi 182, Lucknow 163, Bangalore you see 310, Hyderabad, Mumbai 106, Chennai 177. There are many places. Actually, the last year, the environmental preference index ranked India 174 out of 178 countries for air quality. WHO survey last year found that 13 of the most polluted out of 20 cities in the world in India. So that is the present status of air quality index in India in many places. These are my major pollutants, air pollutants. Suspended particulate matters divided 3 types. Total suspended particulate matter, PM10, PM2.5, one more is PM1 is there now. And sulfur dioxide, nitrogen dioxides. These are major 5 air pollutants in air quality measurement. Even ground level ozone. Yeah. Here I want to say my research part, methodology, how I got this PM, how I will get the gaseous air pollutants, what are the devices, and what are the equipments which are available in our department. We have sponsored it. Okay. So these are all we have in these devices to collect total suspended particulate matter and also PM. Is automatic. You can say automatic sampling for 24 hours. Continuously, these devices working in many places. We have 3 to 4 devices like this. We have identified in Mysore 3 zones, and every day running these instruments to collect this PM, TSPM, PM10 and TSPM. Okay. So this is supplied by Envirotech. It is sponsored in the Department of Science and Technology, which we are keeping in many places. We are collecting the total suspended particulate matter, and also we are collecting PM10 continuously for further analysis. Okay, this is how it works, how this air sucks, and also how it flows and how it collects the particulate matter only, PM10 in a filter, and total suspended particulate matter at the down sections. So this is the outlook of what we collected the PM. If you collect the PM, it looks like this. See, in borosilicate bottle, it is a black carbon. And if you go for electron microscope, you will see like this, the higher magnification, this particulate matter, that is total suspended particulate matter, and PM10 it is. So how you will get it, this means, so this is the filter paper, Teflon filter paper we are using. So this is the white one is a new paper. After partial completion, there is a center one. And complete converting a white into carbon level, black color, that is the loaded particulate matter PM10 it is. So this way you can see this. So today, if you keep 1 day in urban areas, the white paper converted into black paper. Only 1 day is enough. For 24 hours not needed, even 12 hours if you start from morning 10 to 5:30 in suburban stand area, definitely you will get it like this. 1 day is enough to get this type of, this much of PM10, which we are collecting every day. And this is 2.5 sampler. Same Envirotech sponsored by Department of Science and Technology to collect this 2.5 particulate matter, PM 2.5 samplers. It is also automatic, and also it can collect more information, even data also you will get it in this. We switch on this in urban areas, continuously we will get this PM 2.5 like this it is. So this white one is like a control one, the first one. So after 1 day in urban, it depends on zones. Highly urban area, semi-urban area, low urban areas, or residential areas. We have divided like this. So continuously these missions are working in all the places in and around Mysore. We are collecting this PM 2.5, PM10, and also PM1 also. But we are concentrating PM 2.5 and also PM10, which more important to do further research on this. Okay. And addition to that, gaseous air pollutant, sulfur dioxide, nitrogen dioxides, and ozone. Each one requires a separate absorption media. Based on that, we are preparing the protocol. Based on that protocol, we are conducting this. We are collecting the gaseous air pollutants in some places where it's more the emission taking place. Some industrial areas or indoor, we are collecting this. This also gaseous pollutants. I think this mechanism, you can read it, how this NOx forms, how the SOx is going to form, how this acid rain will form. Say acid rain is a combination of sulfur dioxide and addition of NO2 to form a sulfur dioxide. And sulfur dioxide again oxygen to form a H2SO4 in the presence of atmospheric humidity to form HNO3 and also H2SO4. What H2SO4 you can call it, that is called acid rain. Where if sulfur dioxide concentration is more, where the industrial area or fertilizer areas, this acid rain will form, and acid rain the pH is 6. How this photochemical smog, combination of NO, oxygen, and NO2. So water particles, solar radiation to in the combination of HNO3 to form the photochemical smog in a NO2 plus O2 plus HNO3. This mechanism, how the sulfur dioxide is formed. See, sulfur plus oxygen, sulfur dioxide plus humidity to form H2SO4. A natural process. Volcanoes provides 67% sulfur dioxide distributed all over the globe. Means 33% the fuel combustion, transportation, and coal-fired power stations. These are all the reactions, continuous chain reactions to obtain this acid rain in many places. This gives rise to the phenomenon of acid rain. You know the properties of this sulfur dioxide, colorlessness. I'll skip because I want to go the photoremediation concept. I'm skipping this majority of the slides. Nitrogen dioxide, how it forms. Nitrogen plus oxygen. You will get oxygen NO plus NO plus oxygen to form a NO2. The second reaction also favored at the temperature of 1100 degrees centigrade. So you will get this ground level ozone also and NO2 or NO2. Yeah. After collecting all this is the DST project. After collecting the information, after collecting the samples, investigating the toxic effect of particulate matter of air pollution from various emission sources on lung injury and insulin resistance, the mitigation by antioxidant therapy. So we have PM10, we have PM2.5, we have gaseous pollutants, even we have PM10. After characterization of all this, what we did is investigating the characteristics of this particular area of particulate matter, and after knowing the characteristics of that, so which we are conducting on an animal model, what may be the impact on lungs after inhaling this particulate matter. We are using this mice model. What are the impacts which we have analyzed major work on this. Why we are looking on air pollution? Why we have to stick on to the air pollution? Why not in water pollution? Why not in soil pollution? See, on epidemiological studies have estimated 4.2 million people died due to the exposure of particulate matter air pollution in India every year. The studies use mass concentration of this PM as a variable to derive associations between PM exposure and health outcomes. This model of analysis assumes that PM between cities, between countries, are health risks or toxicity per unit dose independent of source and composition. So which is far from the truth. The data analysis from the pooled epidemiological studies have concluded that particulate matter emission sources contribute to more deaths than other sources. That means than other gaseous pollutants. That means particulate matter is the one, a major factor which impact on human health, which impact on plant health, and also which impact on other animals. So this evidence underscores the urgent need for systemic toxicological evaluation of specific emissions of PM10 types in urban settings, which contributes to higher morbidity. That is why we are focused or we are concentrating air pollutions, particularly particulate matter. These are the objectives we have proposed for Department of Science and Technology to get the result. These are the major objectives. One is collection different sources, another one is the ranking characterization of the particulate matter, and to investigate this antioxidant properties by using this N-acetyl or cysteine to mitigate the impact on mice lungs. Okay, one is collection, one is characterization, one is to see the oxidative potential rate of that PM, how much ROS can produce in which place of PM. Okay, so after that is after administration, is there any antioxidant property to control this impacted PM? If you induce N-acetyl cysteine or if you induce any plant extract, okay, any plant extract or N-acetyl is a chemical or natural method by plant extract administration giving this after exposure, is there any impact to release this, or if there an impact to come to the original place of that the animal which are exposed to the PM. After exposure, we have done two ways. One is by chemical method, N-acetyl cysteine mitigation, and another one is plant extract media. So giving plant extract media, we got more effective, significant result than this chemical method. So that means the plants have the more potential, genetic potential to reduce the lungs risk after exposing this PM. That we have proved. We also submitted to the report to the DST. And these different sources, I think I will skip this information. Yeah, see, this is the way which we are going to collect the PM from different sources. For example, agarbatti. For example, charcoal if you burn. For example, cow dung if you burn. For example, mosquito if you burn. Okay, we are all doing this. Mosquito coil we are putting. Agarbatti we are putting in puja time. And also charcoal we are burning. And also cow dung also we are doing for warming. And wood. So all this we are collecting by using this aerotech devices in different chambers. Okay, this is different source of this. So this one is cow dung, incendic stick, and also charcoal and mosquito coil also. Apart from industrial air pollution, we have also done this four important material which commonly using in the publics, which are commonly using in our daily life for many purposes. So this is the redox imbalance, and you see how this injury takes place if the PM enters into the body. Initial injury, redox imbalance, ROS producing reactive oxygen species are generated during the mitochondrial oxidative metabolism, as well as in cellular response to xenobiotics, cytokinins, bacterial invasions. So oxidative stress refers to the imbalance due to the excess production of this ROS or oxidants over the capacity of the cell to mount the effective antioxidant response. Okay, so once enters the production of this ROS is more, so that may be react with many ways. Inflammatory cells will produce, and TGF deactivation takes place, and ECM fragments and pulmonary fibrosis will form if continuously exposing this PM into the animal lungs. Okay. And after that, we did by taking the samples of mice, we did DTT assay to measure the potentiality of this oxidation of the PM. Okay, and DTT assay procedures, these are all procedures which we are following, and this is the ROS rate which we are getting. See, you are getting mosquito coil is high compared to agarbatti, charcoal, and cow dung. Mosquito coil, say how much it is, all the places. Okay, see, mosquito coil producing more ROS than charcoal, then agarbatti, and cow dung. And ascorbic acid to see the oxidation rate to measure this ascorbic acid assay also we did. And ascorbic acid assay is particularly sensitive towards the mosquito coil. Immunoassay we did in the mice samples, even blood and also the lungs BSA fluid. So this is the schematic representation of immunoassay. Overall goal of this assay is to assess toxic nature of PM by measuring oxidized lipid in the human serum, that is APO B100 in the 96 well plates. The results of immunoassay. The immunoassay we observed that mosquito repellent coil has increased oxidative potential compared to the other three sources. That means mosquito coil is producing more ROS than other three. That means more potential to producing this ROS if enters into the lungs in this mosquito coil. This one is the lung injury ranking objective 3. And we did glucose tolerance test to insulin tolerance test also in the blood samples. And these are all what the procedures which we are following after exposing this into the animal model. This I will. These are acute study, chronic exposure also we did. We are on subacute study, three batches we did, and acute study administration PM 24 hours. Subacute study 2 weeks to 2 weeks we are going to do, and chronic study at least 1 month or more than 1 month we are going to conduct the batches like this, and after that we will collect the samples and analyze further. And objective 3, the N-acetyl cysteine attenuates. So these are the results which we got. I'll show you that. Then I want to stick on to the further work on this. Okay, oxidative damage, apoptosis, and comparison. Okay. Yeah, these are the results which we find after doing this. The oxidative potential of indoor collected TSPM by conventional the cellular assay, DDT assay on TSPM. Okay, so these are the results which got after continuous chronic, subchronic, and also acute studies. I think this and all the experimental work which we did after characterization. Okay, this is the point where we are. So that part is first part. First part of my project DST, collecting air samples, characterization of air samples, how it impacts, how remedies by administering the plant extract and also this NST. So now the Center for Photoremediation, sponsored by Vision Group of Science and Technology, Government of Karnataka, has given 48 lakhs to initiate is there plants or potential to absorb these gaseous pollutants or this particulate matter. The concept is same. The plants are any plants are capable of absorbing this gaseous or volatile or this particulate matter. This is the first project which we are getting for phytoremediation process. Center for Phytoremediation we have started, sponsored by Vision Group of Science and Technology, Government of Karnataka, 23 to 24. Now it is also running this work. The scheme is Center Innovative Science and Tech Engineering Scheme. In this area, these are the models which we have developed. These are the models which we have developed. The plants, keeping the indoor plants or keeping the naturally available plants, keeping the potted plants inside a control and also experimental. Is there any potentiality of the plants to absorb this? Okay, so this is the Vision Group of Science and Technology sponsored. Plants have the potentiality to absorb all our indoor air pollutants and also gaseous air pollutants. Okay. So these are the plants we have identified. The objective will test the plant selected from objective 1 at homes or industrial setup. We will select 20 individual homes and apartments in zones of high pollution, particularly close to high traffic areas, roads, after taking consent of the home people, or after taking consent of the complex people or the owner of the house. Okay, so then we will place these monitors. We have the sensors, nearly 10 sensors we have purchased from Vision Group of Science and Technology sponsored project. So that is, we will place the monitors for sensors for PM analysis, NOx analysis, VOC analysis, SOx analysis in hall and bedrooms. Okay, for measuring the baseline levels of indoor air pollution levels for 3 continuous days. After 3 days, the specified number of potted plants calculated based on living area will be placed in the home for defined time, 1 to 7 days. Okay, the level of PM we will measure. The level of NOx we will measure indoor. The level of SOx we will measure and volatile organic. So individual sensors we are placing after taking concern from the house owners. Okay, so similar design will be used as a biofertilizer industry. That is one industry. We have collaborated with that industry, fertilizer industry, chemical industry, located in Mysore, who has consented, okay, to participate in this study also. So that means we have purchased many sensors. We are keeping the sensors in many places. We are recording these levels, and after that, similar way, we'll also keep these plants, selected plants. Is there any potential to observe that level you can decrease the level of that gaseous pollutants if you before placing the plant and after placing the plant in a confined areas, okay, or in a controlled areas. So first we will say selecting one house, keeping all the sensors, we will record the NOx, SOx, VOC and PM for 1 week. After that, we will keep the selected plants. These are screened plants, keeping inside, and also we will place the sensors. We will record the level of PM and NOx and SOx. So that means you will get the comparison, and also you will get, is there any plant potential to reduce this gaseous air pollutants, to observe this gaseous air pollutants? Definitely we are getting a very good result on this. Okay, so this is VGST project sanctioned, which we are running. Okay, so these are the identified plants which we are placing inside. Okay, and recording all these levels of NOx, SOx, VOC and also PM. So first part project is characterization and also impacts and remedies by extract. The second plants are able to do this. Plants are a potential to reduce these gaseous pollutants and also particulate matter, which we are running this, sponsored by Vision Group of Science and Technology. These are the identified plants. The list of indoor plants which will tested in our initial experimentations, not 100% significant, but initially we have screened. These are the species, scientific names. These are species initially identified, which we are keeping and we are monitoring, but not that much of effective selection. Each one, it will take 1 or 2 years to get the out, to select out among this which one is more potential compared to this. So that will another 1 year we will take. Yeah, that is second project. Photoremediation sponsored by Vision Group of Science and Technology. Okay. Vision Group of Science and Technology given that amount to identify plants to screen the level. But this is a 1 crore project. Recently we got it. In 1 crore project is the phytoremediation of indoor air pollution in urban homes to improve the air quality using innovative sustainable technology. That is more innovative sustainable technology, which that is the basic ideas, basic concept, the previous project. So after that, we submitted this. So we can develop some innovative technology, sustainable technology based on the plant wall air filtration system. So sponsored by Indian Council of Medical Research, Government of India, 1 crore project it is, in collaboration with JSS University and also myself, which we are doing this experiment. We have appointed 4 candidates. They are working continuously to develop this innovative sustainable technology based on the plant wall air filtration systems, which we are doing. Okay. So this is the background, the summary, the photoremediation. So rising indoor air pollution in India, homes, offices or schools poses a serious health concern because people spend 80% of time in indoors. Long-term exposure in air pollutants is linked with development of or exacerbations of respiratory and heart diseases and significantly affect the cognitive performance in children. Currently, air purifiers are used, but it is less effective in homes located in high polluted zones. Not affordable by most, hence there is an urgent need for innovative, sustainable, energy efficient and affordable technology to improve the air quality. So this is our major aim or background to keep this, propose this project into the ICMR, Indian Council of Medical Research. They have accepted and they invited for presentation. We did presentation and also they have sanctioned 1 crore rupees to conduct this indoor air quality. The novelty and innovation of this project is we are proposing photoremediation as a sustainable technology to improve air quality. And we are developing and evaluating innovative technology come product plant wall filtration system that couples a passive air purification by rigorously characterized indoor potted plants and active filtration by dust filters. And the next innovation is the effectiveness of this technology to reduce indoor air pollution and prevent adverse respiratory effects of indoor air pollutants will be evaluated in the laboratory after exposure and urban homes in Mysore city and also Bangalore. That previous project only Mysore we are concentrating. This 1 crore project we have to do in Bangalore also. The same way indoor air quality and sensors using this. The objectives to screen and identify superior indoor plant. That's what the previous is only screening. Now identifying superior indoor plant, which one is more potential to purify indoor air pollution, PMO, VOC, NOx, SOx, using experimental diesel exhaust exposure chamber, and to evaluate this efficiency of multi-species of air purifying superior indoor plants, selected plant. Okay, and assembled, second one is assembled a very fabricated frame modular, a plant wall air filtration system. I repeat it, a plant wall air filtration system to mitigate indoor air pollutants using experimental chamber. And to evaluate efficacy of the plant wall air filtration system to reduce the adverse effects of indoor air pollutants in model organisms. That is using Drosophila and also mouse model in diesel exhaust exposure chambers, and finally to conduct field trial a plant wall filtration system to test the efficiency to reduce indoor air pollution, to improve the respiratory health, and to improve the product acceptability in homes and apartments located near high traffic roads in Mysore city and also Bangalore city. That is our the goal in the future, which we are going to show this. Okay. Yeah, this is just introduction. I'll skip this. This also the introduction of what I told many lot information. This introduction part I'll skip. Yeah. So this is the sustainable development technology to prepare a air filtration wall, air purifier wall. The plants are natural purifiers of atmospheric air and use of plants to purify indoor air in emerging technology. So that means it is a filter bed inside. Outside there are plants. Inside machines are working. Inside sensors are working to measure what kind of a plant to absorb how much potential capacity to absorb this gaseous pollutants even PM. Okay, so here the initial studies from NASA and later by other researchers from Western world have demonstrated that certain indoor potted plants are highly efficient in removing and filtering VOC, volatile organic compounds, NOx, SOx, and PM from the indoor settings. For example, a potted, Camidodera elegans removes 60 to 90% of formaldehyde from enclosed chambers. See, that species, formaldehyde 60 to 90% it can absorb and it can remove it. If you place it, that plant in indoor. And Chlorophytum comosum, you can call it as a spider plant. Okay, absorbed a PM very efficiently and reduced the PM levels. Okay, The researchers from Western countries have explored a native plants of their respective countries and has established proof of concept that certain plants have greater capacity to remove indoor air pollutants. This is reported. This is reported in Western countries. Okay. There has been no systemic research carried out in India to identify native indoor plant capable of removing indoor air pollutants. That's why we are proposing. That's why we are concentrating this. So what are we proposing here? To develop and evaluate customized plant air filtration system from time. We submitted the concept proposal by consulting engineers. We have developed a prototype model of the appliance. The following features: inbuilt container for placing and replacing indoor potted plants. That is inbuilt container. And technology combines passive indoor air filtration by plants and active membrane filtration to effectively remove the diverse PM and gaseous pollutants. And a small exhaust plant to pull the large volume of contaminated indoor air. We will also place exhaust fan to pull large volume of contaminated air through the plant surfaces, soil, and membrane for efficiency filtering. And the next is easy. The drip watering to low cost maintenance requires low energy and gives aesthetic look. So this is the model of the air filtration system. The overall goal of this is our project will involve screening known and new indoor plants, selecting indoor plants with high efficiency to remove air pollutants. Assemble the potted plants and the plant air filtration appliance. Evaluate the efficiency to reduce air pollution in indoor chambers. Evaluate the efficacy to reduce adverse health effects of air pollution in mice and Drosophila housed in the chamber with or without filtration system. With filtration system and also without filtration system. And evaluate the efficiency in homes located near high traffic zones. These are the overall goal of our work after developing that model in Mysore city and also Bangalore city in selected areas. These are all indoor air pollutants, different already. You can also Google this. Last line is there. No? You can search it. You will get a lot of information on this. If you want to search more detail, if you want to propose a project to the DST. These are all information where we got it, and also this Shah et al. What are the reported that 1% increase outdoor knocks. The references and also places where they did this work is doing in many places, and also they did many reports on this. Yeah, a lot of adverse effects that I will skip this one. Limitations here now. Limitations of air pollution purifying is for maintaining the indoor air quality. Air purifiers with high efficiency of HEPA filters, FA filters, we will call it as FA filters, are proven to be effective in reducing indoor air pollutants, particularly PM in urban homes. However, there are several limitations. Not effective in homes if the windows or doors are kept open. These are the limitations which we are facing and which we are experiencing. And not effective in homes if windows or doors are continuously open. Commonly seen in Indian urban homes. Not affordable by most Many people not accept. Okay, we don't want. If you approach them, no, we don't know. We don't want like this. They are not offering to keeping this accepting keeping this sensors inside the home and high maintenance cost. That is electricity and less effective in homes located near the high traffic zones with poor building mechanical ventilations. And air purifier are switched off during the daytime when the occupiers are outdoor, at work or in the school, and leading high indoor accumulation of air pollutants, which overwhelms the appliance of efficiency and limited capacity of filtration of volume of air. The common man presumes that air conditioners purify indoor air. No, however, it is not true also. AC regulates indoor temperature, but it is not on indoor air cleaning system. ACs. Certain new ACs have built in dust filters to trap the coarse particles and allergens. However, cooling system in AC recirculates the trapped particles inside the room with time. These are the major limitations which we are facing. These are the limitations we are experiencing in many places in Mysore. Similar to this, they expanded the list of indoor plants with high capacity of VOC. The efficiency to remove VOC ranked in the following order: substrate, soil material, siliceous, followed by soil microorganisms, followed by plant foliage, and also plant aerial parts. So numerous studies have demonstrated that plant leaves can directly absorb NOx via stomatal activity and assimilate into the leaf nitrate and ammonia acid. Okay, you can refer these 3 papers. The plants also reduce indoor PM by physically trapping the aerial parts of, especially by the rough surface, lower surface, trichome, the leaves through gravitational sedimentation, particle interceptions, and Brownian diffusions. Govornoska and Baker et al. Reported that leaves, that is Chlorophytum comosum, spider plant, traps indoor PM very effectively. Several leaf traits such as leaf surface, leaf area, and space area shown in the influence of PM trapping activity. You may find this PMID based on the literature. These 4 papers you will get more information. What the ID, PMID we have given. If you search it that 4, you will get how the mechanism of the spider plant absorbing this PM in a different surfaces, upper surface and lower in stomatal region and also the stem regions. That is why spider plants are the one which has the more potential, genetic potential to absorb this PM other than this. Okay. The gaps in knowledge and also the study. The laboratory studies have overwhelmingly proven that certain individual plant species reduce indoor air pollutants, but the effectiveness in urban homes is yet to be investigated. The question: no systematic studies carried out to screen locally or endemic indoor potted plants for their air purification capacity. The efficiency and effectiveness of group of single or diverse plants species with a superior air purifying capacity to reduce diverse indoor air pollutants. There is no products in the market coupling both plants and membrane filters for cleaning indoor. So far, no such type of the market available of this novelty. Okay, our proposed study is novel of several aspects and also highly innovative for improving microenvironment air quality, which was living areas of urban homes. And that is all the laboratory innovations which keep this. The acceptability and sustainability is the following features increases the acceptability and sustainability in our product or technology. The plant wall filtration will be equipped with hypogenic indoor plants and requires very less energy and affordable, easy and low cost maintenance. That is why we have mentioned the title low cost development of the photoremediation. Development of low cost, that is which should not be cost economy or more effective cost on this. Always is affordable to the publics and easy to maintain and easy to get in the market. That is our concept of the future days. So installed in any indoor settings such as homes, schools, offices or malls. Okay, we provide aesthetic look and product may also fruitful enhance the efficiency of existing mechanical ventilators. This we have mentioned from one by one. So that the law is to conduct field trial, the plant wall filtration system to test the efficiency, the indoor air filtration or indoor air quality improvement or improve the respiratory health and the product acceptability in homes and apartments. This I have done. I have shown this. Yeah, this is the work procedure, generation of vehicle emissions, approach, spotted plant indoor. Yeah, so this the commonly available and listed plants which we are conducting the experiments, we are keeping this inside the chambers in our laboratory area and also the urban areas. So these are the common names and scientific names which continuously we are working on it. But we have started recently, last 4 months back this. We need more time to get screening of this in an effective way in the laboratory wise and also the homes and also in the commercial city also. So this object, evaluating this, how the work plan of this chambers is to evaluate the efficiency of the multi-species, purifying the superior indoor air plants, assembled and fabricated, framed modular plant wall filtration system to mitigate indoor air pollutants. Okay, using exposure chambers, okay, simultaneously living and also office rooms. A plant wall air filtration working principle is we have designed and prepared a prototype model of a plant wall air filtration unit. You can see the figure 1 to 3. So which will hold multiple plants from virtual plant foliage wall with beautiful aesthetic element to indoor living space. It has to look nice. The features of working principle of this unit is as follows. So designed to purify indoor air by coupling both passive and also active filtrations. You can install the passive filtration through potted plants and active filtration by membrane filters. Okay, both passive and active filtrations. Then separate containers to load and replace the potted plants. So we have fixed separate container to replace the plant and also place the plant. And up to 9 potted plants you can fit in a single the filtration system. Okay, the leaves of the plants are arranged to form a virtual leaf wall. The soil of the platted plants will be covered with the activated carbon pellets to absorb the VOC. And dust filtration fitted rear plant wall will filter the coarse particles. And activated carbon sheet will absorb VOC. And electrostatic filter will trap the fine and coarse particles. And low dust axial fan will pull the dirty air inside the unit and release the clear air inside the room. The dirty air will be pulled through the potted plant wall and dust filter followed by chamber, and electrostatic filters which clear the chamber, and the seventh step is clear air relief. So water tank for drop watering plants. So this setup we have done it. You can see the assessment of air purification by plant wall filtration system in a large exposure chamber, simulating in a living room. The fabrication work we did, and the length, width, and also height we have made aluminum framed glass partitions and simulating the smaller office and living spaces inside the laboratory. Temperature controlled conditions we are maintaining, and the out duct will be connected with HEPA filters before releasing the dirt air outside the laboratory. The airflow inside the chamber will be controlled. And testing plant air filtration unit, and with and without plant. Experimental design told in the beginning. Every day, first we will place the group 1, group 3, group 2, the respective chambers. Group 1 is one type of plants, only one category. Group 2 is another type of plant. The same species of 10 plants. For example, spider plant, only 10 plants will keep in group 1. And the snake plant only 10 plants will keep in one filter. Okay, group 1, group 2, group 3, with respect to chambers, to export the ambient air for 3 hours and transport them back to the animal house, holding room. Subsequently, we will place group 2, group 4 respectively in an exposure chamber and expose them to the diesel exhaust for 3 hours. So we'll see the exposure without planting, and we'll see the after placing the plant, we will measure the level of this by using these sensors in this PM, NO2, CO2, and also other continuously. So after this assessment, we'll also place the animal and the Drosophila. The similar way we will assess the concentration of this PMs. This animal model experiments this. Even Drosophila and mice model we will do. In the last year this is. Yeah. So our study will develop rigorously tested working ambience in a plant well activity or air filtration unit. We anticipate our field study in Mysore city and Bangalore will demonstrate the appliance of plant wall air filtration unit will significantly reduce indoor air pollutant levels in urban homes, apartments, and traffic areas, and also the respiratory infections among the home residents. We expect that the developed appliance plant air wall filtration unit can be used in offices, schools, and also theaters, malls, or sports complexes, or gyms, etc. Okay. So that is our third project objectives, methodologies, design, and future goal. First one is characterizations. Second one is screening the phytoremediation. Third part is specific model, air filtration model, and giving more importance on screening a particular type of plant. Okay. See, as I explained, or as I mentioned, not that much easy. If you conduct the experiments, it will take years. But 3 projects are running. Continuously, we are getting the good result and response, and also we are working on it. Many students are working on this. Okay. Then in general way, technology that use the plants to clean up contaminated sites. Yes. Green technology that uses the plant system for remediation and restoration. Yeah. Not only the air, you can restore, remediate the soil. You can also remedy in the water pollution areas. Okay. In a general way, encompass microbial degradation in the rhizosphere as well as uptake, accumulation, transformation in the plant systems. In the plant systems. So this has broadly classified this technology, phytoremediation technology. Okay. In 5 major areas. One is phytoextraction, phytostabilization, phytotransformation, phytostimulation, phytovolatilization, rhizofiltration. So in these 5 ways, we can remedy using the plant in different ways of extraction. Hence, the phytoremediation broadly classified: phytoextraction, phytostabilization, phytotransformation, phytostimulation, phytovolatilization, rhizofiltration. Okay, so phytoextraction, the roots absorb the toxicants or pollutants, which can enter into the shoot system and releases into the atmosphere. Less toxic. Stabilization, the plants can absorb the toxicants, or plants can absorb the gaseous pollutants, and which can store in the shoot pot without releasing into the atmosphere. That is stabilize, immobilize that. Immobilizing this contaminants. That means in the form of absorption, accumulation by roots, or precipitation within the roots. That means phytoextraction is the one, so where the contaminants from roots above the pots absorbing and concentrating and precipitating the contaminants. But stabilization is which will not release us outside. The plant will keep it. Certain point of contaminants, or you can call it the bioavailability. It is there in the plant, but it not released into the atmosphere, which will absorb through the roots, and it form another form and will keep it in the stem region or store region or even leaf regions. That you can call it a phytostabilization. Phytotransformation, which will convert into another form. Okay, and break down into the contaminants into a simple way and taken up in the plant in metabolic process within the plant and remediate some organic contaminants such as chlorinated solvents or herbicides or even munitions. So that means it will transform into other form and keep it in the system, shoot system or root system, or it can address the contaminants in the soil or sediment on the groundwater. Or phytostimulation, that is, break down the contaminants within the plant or within the root or within the rhizosphere, which is with the help of bacteria or with the help of other microorganisms in the rhizosphere. That is phytostimulation. So microbes, rhizosphere transforms contaminant into non-toxic product. Ado phytostimulation. Okay, phytostimulation in a root system or shoot system. And phytovolatilization, yes, which involves plant taking contaminants from the soil or from water or transforming into the volatile forms or transpiration into the atmosphere. Okay, That is called a phytovoltaization. It works on organic compounds, even heavy metals contaminants, and that is TCE, or the metals contaminated. Mercury is major one. That is the voltage it converts and also evaporates into the atmosphere through transpiration. Okay, and the last is that is rhizofiltrations. So rhizofiltrations absorb the or preparation into the plant root system, which absorb the contaminants in the surrounding of the root area. Rhizofiltrations. So that means all parts of the plant, okay, all parts of the plant, one way or the other way, it will transform, or it will volatilization, or it will rhizofiltration, or it will may phytoextraction. So one way or the other way, it will convert a toxic xenobiotic compounds into non-toxic, or it will convert into volatile organic compounds, or it will store in the plant system, or it will store in the root system, or in the leaf system. So that phytoremediation broadly classified into 5 groups: phytoextraction, phytostimulation, phytotransformation, rhizofiltrations, and phytotransformations. Okay, so these are the major types stabilizations. In general classification in phytoremediation process. Will I stop here? Current methods: RCRA, Resource Conservation Recovery Act, landfill, a pump with the treat of the systems. I think time is over. Hello?
Sir, time is over, sir.
Okay, thank you. This is general information which mechanism that and all you can see. This I told, rhizofiltration, phytofiltration, and also phytotransformation stimulation. Advantages, a lot of advantages you can, okay. Even aquatic plants, there are many aquatic plants are chosen to absorb the particulate nutrients and also remove the pathogens, metals, and also other contaminants. There are many hydrophytes. Hydrophytes again classified into 5 groups. That hydrophytes are play an important role to clean the water, fresh water and also marine water. These are types of hydrophytic, and the root system, the contamination removal, chemical, physical, and also biological methods. Rhizofiltration applicability. Yeah, the rhizofiltration, I told you the cadmium or mercury or heavy metals it will absorb. Limitations are many, and example of experiment that we are developing. Okay, although much remains to be studied in phytoremediation, will clearly play some role in the stabilization and remediation of many contaminated sites in the future research. The main factor driving the implementation of phytoremediation projects are low cost with significant improvement in sites, aesthetics, and also potential for ecosystem restoration.